Hi everybody, welcome to our first vodcast in our cell unit. In this vodcast, we're going to spend some time really getting into some of the early distinctions from one cell to the other, start talking a little bit about some of the basics of the cell. So let's dive right in. Cells are our basic unit of structure and function for life. All living things are made of cells. As a result of that, we, just like the atom is the basic unit of matter, right? Of all the stuff we're made of, we have the atom. For life, that basic unit is the cell, and it is a very small structure that has a lot of working parts in it. And by studying cells specifically and what those cells are, how they work, we can learn quite a bit about how organs and tissues are the way they are, how they work, based on the cells that make what they are, that make them up. And, just to kind of give a sense of this, we know that there are all types of life, but one of those things we don't spend a lot of time on are viruses. And we all know about viruses. It's flu season, right? So people are getting flu shots, and that's caused by a virus. But the thing about viruses is that we're not really sure whether we consider them living or not. And we'll talk more later down the road about why that might be, but viruses they're a very small unit, but they're made out of protein on the outside, and on the inside they have a form of heredity information, so genetic information, either DNA or RNA. And so here I have a picture of the HIV virus. Okay, so this is the virus that causes AIDS, and this virus is on the outside of a helper T cell, which is in humans. It's our helper T cells are a part of our immune system. They are what help fight, the bad things that come into our body off. So when a virus infects a cell, what they actually do is land on the outside, and then they inject their genetic information inside. The virus itself never actually goes in. The whole thing never goes into the cell. It's just the genetic information. And then that information basically takes the helper T cell over, and it becomes this virus-making factory. So because of that, and they're not technically a cell, that's why there's a lot of debate as to what to do with viruses. So I figured, we just kind of throw those in there just to give you a little food for thought. Now, cells. Since they're the basic unit of life, they've been around as long as life has been around. But we as humans haven't always known what's made us up. So it all began way back in the 1600s with a fellow by the name of Robert Hooke. He was the first scientist to actually view the cell. So he took a piece of cork and sliced it very thinly and placed it underneath the lens of a compound light microscope. Not too unsimilar to ours. The only difference being, just like this right here, the only difference being that in 1665, we really didn't have electricity yet. So they had to rely on mirrors in the sun to be able to get the light through. And as he looked through this microscope, he saw this right here. And what he saw were these very square-looking, organized structures that looked very empty. So, as a result of them looking like square, empty objects, he called them a cell, because at the time, a small, empty room was known as a cell. So that's where the term comes from, and since then, we have always called it that. And we know now, boy, they are not empty at all. They are filled, more filled than even I can really comprehend and even understand. They are so jam-packed with structures that work on a, such a fast level that cells are truly amazing. So let's work our way up. So that's when we first discovered that living things were made of cells. And so this is the first part of what we call the cell theory. There are 3 parts we're going to learn to this. And Robert Hooke gave us that first part, that all living things are made of these very tiny units. The second part to the cell theory came when 2 scientists by the name of Schleiden and Schwann, almost 200 years later, so we have a pretty big gap right now, where they were looking at different types of cells, not just plants, not just the cork like what Robert Hooke looked at, but at animal cells, plant cells. They were looking at objects from all different types of organisms, and they found that cells existed in all of these. So it sort of backed up Hooke's statement that that life is made of cells. And then through the advent of staining, they found they weren't empty rooms at all, that they were filled with structures that began to show up when they started using stains to observe these cells. And they found that cells made up the basic unit of life. There wasn't any structure that was universal, that was smaller, that could exist on its own as a form of life. So almost 200 years later, second part to the cell theory comes into play from Schwann and Schleiden, and it is that cells are the basic unit of life. We go not too far more than that, and we have Rudolf Virchow. And Virchow's contribution to the cell theory is a pretty important one, but it's this contribution that keeps the cell theory a theory and not really law. And that's because his statement was that cells come from pre-existing cells. The other term is to beget, that cells beget cells. You can't just throw a bunch of ingredients into a flask and voila, make cells. That cells exist because cells divide and come from a pre-existing cell. So in essence, life comes from pre-existing life. Makes sense. We understand that. Problem with that is, let's go way back in time, and there's a really, really big, important question we have to answer, and I'm sure you're probably already starting to think about that question, and that is, so where the heck did the first cell come from, right? So if we, scientists are thinking about that, there's lots of theories out there as to where that first cell came from. But sadly, none of us were there at the time. So it's, as we work towards trying to figure that out, that's what keeps this as a theory. So just to review, the three parts of the cell theory are: cells are the basic unit of life, all living things are made of cells, and all cells come from pre-existing cells. Cool. So let's start talking about cells themselves. To do that, we need to get small. Cells are tiny. They are microscopic. We've already started looking at some. So we need microscopes to be able to look at cells, and the better the technology, the more we learn about cells. And there's really three types I care that you know about. There's the compound light microscope. Those are our scopes, the scopes we use in class. And that is what we see image there. That's paramecium. That actually, the picture was taken from our microscope. Then we have transmission electron microscopes and scanning electron microscopes. A TEM is a type of microscope that sends electrons at the image, and when it bounces back, you get a picture of what you're seeing. The TEM created this one right here, and it's extraordinarily high magnification. I'm talking like hundreds of thousands times magnification. It also sends back pretty incredible detail. So we can see structures that we can't see with our microscope. So for example, things like this one right here. This is a chloroplast, and we can see the insides of a chloroplast with a TEM. And then scanning electron microscopes are really cool because they do the same thing a transmission does, but it sends back surface image. So we get almost like a 3D surface image of what these look like. So here we have blood cells. And this is like human blood. So we got to look at human blood. Here are our red blood cells. There's a white blood cell. These tiny things over here, those are platelets. So we get to see what these look like as a 3D, because we sometimes forget these images and these things we look at under the microscope actually have depth and are, are three-dimensional because they look so flat under what we see. And one thing we're going to get used to using, and we started doing with the microscope lab, are the use of stains. Stains are so important, and we saw that in the scope lab. We saw how stains allowed us to see things better. So with staining, the use of the scope, we can really get in pretty heavy into understanding what structures are inside cells and how they work. So classification of cells. Two ways we can classify a cell. I can take all living things and I can bust them up into just two groups based on the types of cells that make up what they are. Comes down to one thing. Do they have membrane-bound organelles or don't they? And a membrane is a dual layer that we're going to talk more specifically about in terms of what it's structured as, but it's a lipid or fatty layer that creates the outer edges of these organelles. And an organelle is a structure inside a cell. So I could take all life and I can say, okay, you guys that have membrane-bound organelles, you go over there inside your cells. If you guys don't have membrane-bound organelles inside your cells, you go over here. Two groups. We call the first one prokaryotes. Prokaryotes are bacteria. Really, that's the first group. If you're a bacteria cell, then you're a prokaryotic. Pro means before. You're going to see this prefixed often. So pro means before. Karyote comes from the terminology in the Latin means kernel. And the first time they started looking at cells with stains, the nucleus would stain really dark. And you could see this dark object inside a cell. And it looked like a little kernel, like a corn kernel. So that's why they called it that. So they determined that if you didn't have that, you must have come before in the timeline of life. So they called them prokaryotes, before the kernel. So these are your bacteria. They don't have any nucleus in them. They don't have any membrane-bound organelles. That part's key. No membrane-bound organelles. That includes the nucleus, because that is a membrane-bound organelle. And their DNA, which they have, because all cells, all living cells, contain DNA, simply just floats around in their cytoplasm. That's what they do. They float around in the cytoplasm, and that's it. So besides having no nucleus, besides having their DNA in their cytoplasm, they have one other major organelle that all cells have this, and it is called a ribosome. And all cells have this because it is so important. It's the organelle that deals with constructing protein. So we'll really get into that as we come down the path into our discussion on cells. So besides that, that's what they are. They have a cell wall often. They are single-celled organelles. No prokaryotic organism is a multicellular organism. All prokaryotes, which are bacteria, they are single-celled. That's it. That's what they are. And sometimes they'll have projections called flagella. You know, they're a little bit specialized in that manner, but otherwise they're pretty simple when it comes to cell structure. So those are your prokaryotes. The other type of cell that all the rest of life, your animals, your plants, your fungus, your protists, the other four kingdoms, all are what we call eukaryotic. And the U part, that prefix means true. So when they looked at the cells with the stains and could see the nucleus on the inside, that little kernel was there. So they called these the true, true kernel they were eukaryotic. And in all eukaryotic organisms, their cells all have their DNA enclosed in a nucleus, this membrane-bound sac that separates their DNA from the rest of the cell. So here would be the DNA inside the cell. Here's where we separate it. They also have a huge number of complex membrane-bound organelles that exist in and around the cytoplasm and carry on very particular functions. So really that's where we're going to begin our look at cells. We're going to spend our time getting into what these organelles are, what their function is, why does a cell have them, because to be honest, cells use these very particularly, and they all have a super important function for the life of the cell. And so the cell wouldn't have it if it didn't need it. So we're going to look at what some of the major organelles are and understand how they work. So there's our opening look at cells, and so I'm pretty excited for this unit. I think it'll be a lot of fun, and as we start to kind of put together the pieces of what makes life exciting. So have a great night, and we will see you guys in class. Take it easy.